Hey there, thanks for joining us at the Rabbit Hole of Research, where you'll learn a few facts that you can impress your friends with at a party.
Hey, welcome back to the Rabbit Hole of Research, down here in the basement studio. For the mini, you have me, Joe.
Georgia.
We've got Georgia.
Nick.
We've got Nick. We're all crewed up for the mini. You know how we do it. A little mini recap of the last episode 74, part 2 of our Spider-Man villain series, Scorpion and the Other Chimeras. And so we had our guest, Aaron C. Anthony. And so she was a evolutionary biologist and science communicator and teacher. And so really cool. It was a great episode.
And I was just thinking before we started recording, but I was just saying, it had a lot of science.
It did.
Which is nice.
Yeah. The superhero ones.
Yes, we were very sciencey this time around.
I was going to say the superhero ones, even last season for the Fantastic Four, the first time we tried this format, we did a 4-part series. They got pretty sciencey in there. I mean, some philosophical at times. So that was.
I do feel like it's because they're written by humans. Like it has that more grounded feel for us, where we all kind of have that bits to relate, and they were just as curious about science as we are.
Or try.
That's where we all dive in.
Yeah, I feel like Aaron brought a lot of that. Obviously, that's a really big passion to communicate science. And
so a few things.
And it's always a good jumping off point.
I was just going to.
I was just saying it's always a good jumping off point for everyone to get into the sciences.
Yeah, it's like the hook them in. There you go. Then you got a little science over here. Look over here.
Science.
Yeah, facts to you. Impress your friends with, right? That's what we always say. So we try to give it to you.
Is that what we always say?
That's what at the beginning of every episode. Or at least that's what I say. I don't know. You guys don't say much on the intro, so.
Yeah,
I just say it's my name.
Yeah, why is that? Why don't we say anything on the intro?
I don't know. You guys didn't take the
I think we need to
intro recording
We need
serious.
to re-record that intro.
I think Nick's in the trailer. I don't know if people actually listen to the podcast trailer, but we do have a trailer.
Oh, yeah. We should revisit that.
Some things to talk about in there. When I went back over and doing it, things that we mentioned. One thing I didn't mention, we were talking about studies and animal studies and things, but where research science is moving away and using something called organoids. And organoids are these kind of tiny, self-organizing, three-dimensional tissue cultures that are grown from the stem cells, right? So in these cells, they can coax it to mimic kind of the complexity and functionality of the actual human organ they want to study. So they can make like little tiny, in the order of millimeters, brain, liver, lungs, to kind of have a model to see how the organ functions and or drugs, or how the drug would affect the model, how would it affect the organ, or doing different things. So it's a really cool system that's been developed and being used now.
I'm trying to picture that, because if you think about the brain,
right,
it has all those different parts and they all have different functions. So are they able to model that all in a little tiny
or different
model?
parts of the brain, right? Yeah, so they can
so
have
they're only maybe looking at a certain section
of the So
and so then they
right. So your study is specific, right? You can't grow an entire organ, and the reason is that the vasculature, and we talked about this in the episode. You have vasculature, you have other structures. The vasculature, which brings blood, oxygenated blood in, deoxygenated blood out to provide nutrients for the tissue. So if you get too big, you need this structure. So for these tiny organoids, you keep them small so that the nutrient bath they live in, you can actually provide nutrients for the tissue, and that's how far it can penetrate in.
So how many
without the vasculature?
If they're little, like these organoids?
Yeah, organoids from width of a hair to about 5 millimeters.
Let's say you're studying, I guess it's all different.
Right.
But how many cells would they typically have?
Yeah, you're probably talking.
You can count those?
Well, you can estimate, right? So I'm guessing tens of thousands, maybe even millions
and
of cells.
And if they're just a hair, then it's probably...
Right, and it depends on what you're studying, right? So I was going to mention, because what they did is on the Artemis II mission where they went up, they had taken organs on a chip. So they took cultured organoids on a little chip dish and took it up into space.
Not like potato chips.
Not like a potato chip, like a business card sized little container,
Wow.
but maybe flat. So it wasn't very big, but you had these functioning kind of mimics of the organs on there. So you take that up and then you see the effect of microgravity and things like that affect the organs. So then they went up, they did their mission, and they brought these back down, and now scientists are taking this and doing their studies and review.
I'm just picturing that and how hard that, because you see those pictures of the astronauts and everything floating around. You can see them. Oh no, I just lost my brain organoid.
Yeah. It's my organ on a chip. There it goes.
Flying across.
I think they're not throwing it around.
Are you sure, Joe?
Yes, you're right.
I feel like they would do.
When they're trying to get people excited about science, they're probably doing this kind of stuff, right? Yes, but I think when they're in there doing their experiments, they're throw me the pipette across the module.
A microscope in space. I mean,
that I don't know.
Because
I'm unsure.
They really have.
Yeah. They could. They
are
like
very tight for
dissecting
space,
scope or things like that. So yeah, that's probably why they're bringing it back to Earth to do more studies. But yeah, so it was really interesting. I didn't mention in there other uses: personalized medicine. The organoids can be tailored to the person, right? So they can use your stem cells to form your cells to form the organoid, and then see how your organ would respond to this drug or something like that. When you
say something like that, specialized to the individual.
Right.
That just, for me, all I see is a giant dollar sign.
Well, yes,
because
that is true.
Is that just mean the people that have lots of money would be able to have customized medicine?
You're right, because now...
Because, I mean, we're in a lot of people their insurance won't even pay for, a certain blood test, let alone I'm going to be able to get a customized.
Yeah, no, personalized medicine is exactly that. That when you take medicines and then there's some dose that you take and they go, well, let's start at 5 milligrams, see what happens, then 10 milligrams, right? Because this was tested on an aggregate of thousands and tens of thousands of people.
Doing their job.
But also, it's not tailored to you specifically. So how your body is going to respond to that, the side effects that you're going to have, it's unknown until you personally take the medicine. So you're right. So here you could have a case where they go, okay, let's take some of your cells, let's grow up a little organoid, and we're going to see how this liver drug affects your cells. How does it absorb it? How does it process it? Is it as effective as the control group? And then come back and go, okay.
You don't have to suffer the side effects.
Potentially, right. You could take it a step further and go, well, after you do that, you could go, well, here's other information we've learned. And so now we've used, let's say, to make some custom drug based off the original drug we had. But we found that if we change this receptor, remember this lock and key, we talked about that in the episode, that we change this a little bit, it fits into your cellular locks a little better and it'll be more effective. Wow, look at that. So that's a personalized medicine. It's just for you. And someone else, it may work, but it might not work as efficiently. So really, really cool. And you're right. That leads to some dystopic thinking there. But then you got disease modeling. And I said drug discovery and toxicity. So those are the things that you use these organoids for. But really, really cool because you get away from animal models. So you don't have to do these experiments in mice or rats or chimps or the other animal models that we use. Sometimes you may have to because if you need to look at the entire organ. But
and you use human cells.
That's right. Yep, exactly. That's right. So it's even more direct. But so that was what I mentioned that. In the episode, Aaron talked about medical research on human research and being paid. And in 1965, Scorpion, he was paid $10,000. And she said, oh, what was that in today's money? You can do the inflation kind of calculator. It came out to $106,000. So it was a lot of money for this test to be done. But so, yeah. And then we started the episode about Spider-Man. Is he a chimera? And it was like, no, because he doesn't really have, his DNA was mutated to create new spider genes per the lore of the comic. But it was really interesting, and I don't want to spoil it, but in Spider-Man, Spider Noir, the Amazon Prime series, the way that they were created was much more chimeric in nature, that they did actually have a fusion of the other animals, the spider's cells integrated, and his powers were even a little more integrated. He had actually the webs were coming from out of him right. So it was interesting
there were some scenes that were really
right. I don't know, we talked in the last meeting about how the Spider Noir felt a little different than the Spider-Man that maybe we're used to and familiar with, but that was something I noticed. We went through the episode and I was like, oh, you know what,
interesting,
it was a little change.
Unfortunately, I have yet to finish the Spider Noir series. As I like, I don't. I'm shocked.
Yeah, I understand your.
Yeah.
Especially when you've read the comics and you know the stories, and then of course the adaptation.
Right.
That always makes a difference. And I had not read the comics.
Yeah.
And I loved the series, but it was based on just the series for me. So.
Yeah. The other thing that we, in the episode, you had referred to a mule as a Jenny.
I was just about to bring that up.
Oh,
I was just about to bring up.
Look at that. We're there. It was interesting. And a listener commented, I want to say on Spotify, or I think it was Spotify.
Yeah, it was on Spotify.
Yeah, and I had looked this up. And so, yeah, Jenny is a mature female donkey. And when a Jenny is bred with a male horse, a stallion, the offspring is called a hinny. If you breed a male donkey, which is called a jack, with a female horse, which is a mare, the offspring then is a mule. So they have all these names, all these
That's
different
awesome.
animals.
Yeah.
But yeah, so a Jenny is a mature female donkey, and a jack is a mature male donkey. And then you have a male horse, the stallion, and then you have a female horse, the mare. I was going to say the Italian stallion, but then we know that's Rocky. But
Yeah, I don't see us going down.
No, we're not. Yeah,
not right now.
Not right now. Maybe season X. Yeah. Then there was some feedback about the splicing. One of the questions in the newsletter that had posed was if you could pick an animal to be fused with as a chimera, and Alex, he said the wolf, and he would become a werewolf, a real werewolf. And then we had some banter back and forth about using gene editing to get the best qualities of the werewolf, but not the insatiable appetite and loss of kind of control. That's right.
You can't really have your cake in.
Well, like we said, it comes down to calendars. That's right. Yeah, it comes down to calories. So how many Big Macs?
And I just, when you talk about that, all I can picture is Jack Nicholson.
Yeah.
Jack Nicholson. Oh, wow.
And Wolf.
And the wolf.
Yeah. Was it just Wolf, right?
Or
was it the Wolf? Okay,
I think it was just Wolf.
Yeah. You had a medallion and stuff,
I
right?
did. I had a medallion because I went to the opening night when that came out.
Yeah.
I don't
That's
know.
funny.
I don't know where that is.
I don't know either. Alex also sent a link to Grand Theft Auto 3, I believe. It was a wiki about the man in a trench coat. I guess there's a whole fandom about some man in a trench coat who turns out to be a serial killer. So that fits Nick's theory.
How well do we know Nick?
I know. I
mean, I'm not the one out here in trench coats. I'm like someone that has openly said they wear underwear.
I don't know who that. I don't wear a trench coat. Yeah.
Yeah, sure, Joe. But
yeah, I was going to then. Go ahead.
Do you know what kind of animal you'd be fused with, or did we say that on the show?
I think we did say it on the show. That. See, if you would have listened again.
Wow.
So listen to episode 74.
No, you know what, out. Nick? I was sitting here going, I can't remember what.
Yeah. And also, I think I mentioned that in the newsletter. So if you're at the newsletter.
I have to say, Joe, I really enjoyed just looking and reading the newsletter. I think it's really well done. Yeah.
Well, thank you. We're getting a little bit of assistance on
Wait, we have a newsletter?
Yes. All right. You guys go out, subscribe to the newsletter.
Notes.
Yes, the show notes. But yeah, it comes as the episode's released, you'll get an email and it has a digest of what we talked about in the episode. Kind of
I like that.
Some science terms.
I like that you break it down. Here are those things that you could use to impress your friends. Right.
So there's the fun facts in there. So I'll harvest those. That's a few. There's probably many more, but just some really cool ones. But if you're not subscribed, go subscribe. And then you also see what we're up to. You can kind of keep in touch with us. So, yeah. So go subscribe to the newsletter on Substack. But,
see, that was such a good way to organically dive into that one.
Yes, that's right. Yeah, go support us, please.
We don't do this often.
No.
Sometimes we need to ask for some support.
Yes.
Nice.
Yeah. Anything else before we move on from the episode?
No, I thoroughly enjoyed this episode. Yeah. Actually, this whole series is just right up my alley.
It's really good. Yep. We have one more. We're going to finish it out, part 3, with Alex Haynes. So that was a fun conversation about the influence of Spider-Man's villains on Spider-Man himself and how we feel about Spider-Man becoming a villain. So that's a little tease in there. We went down that rabbit hole. But yeah, check it out. It'll be out in a couple Wednesdays or next Wednesday.
And,
after you hear
again, I think we have a lot of new listeners on this episode too. So it's greatly appreciated to see everyone finding us now, and hopefully everyone goes through our old episodes.
Yeah, definitely. We alternate between a full episode and the mini here that we do a little recap, and then we add the science holes, touching on some current science, and then we do the media that we're digging and consuming and what we like about that, to give you guys some long format media that you can dig into and join what we're doing. So cool. Thanks.
All right.
Now we need a little jingle. Science holes.
No.
Science hole.
Right, there you go.
Look at that hole at the science hole.
All right, maybe not. Well, we're going to get there. We're going to have something figured out for a little transition. But yeah, you guys, somebody want to start? I've been talking for a while. Like I do all the time. So.
I'd read Jordan had something, right?
That's right, yeah.
I did. So right now we're having this, I don't even know. I don't know what to call it, but it's not like a pandemic. It's not like, but
outbreak or endemic. Endemic, I think.
So that might be something I'd love to know. That,
right. Yep.
What are those terms? But a lot of people are getting sick, and the big thing that's happening is explosive diarrhea, and I think people feel bad for about 14 days. And to be honest, this has really been freaking me out because I went out to a festival and then I got some tacos and I had cilantro and onions on my tacos, which why wouldn't you? Then I remember, they said cilantro and fresh herbs is one of the things you should avoid. So I'm eating this. And then in the middle of the night, I'm waking up and I'm like, my stomach is upset. And I'm thinking, oh my God, I have this. And luckily I felt better by the next morning. But I guess I just like a little more science on it.
Yeah, just some facts. So it's the Cyclospora. It's a single-celled organism. It's not a bacterium, but it's actually a protozoan. So it's in this other kingdom. So you have kingdoms of organisms like bacteria, fungi, planta, animal.
And it's a parasite.
It is a parasite, right? Yep. So,
and so what makes something a parasite? When it can only live if it's on a host?
So it's an organism that lives on or inside another organism. Gets its food at the expense of a host. So
and I think just hearing that word, parasite, and you said it's a single cell.
Single cell. So it's a single-celled organism.
But if you were to get sick, you have more than just one single cell.
You have more than one.
If you only have one little tiny single cell.
It's not just one. I was describing the organism itself, that it's not multicellular. So, something, you are multicellular.
Right.
So you exist as many cells, and that makes you. But this thing can exist as just one cell. So one cell can grow and form.
So let's say, you know how you have probiotics in your stomach?
Yes.
Are those single cells?
Those are bacteria, and most are probably single bacterium, yes. E. coli, salmonella, those are single-cell organisms, but they're bacteria. This is a protozoan. So protozoans are eukaryotes. So like our cells, they have a nucleus, they have mitochondria, so they have a thick cell wall, so they're a little harder to kill.
Right, and that's
so it lives
like...
Can this go to animals too, or no?
You mean the Cyclospora itself? I think humans are the only known hosts.
And it's in 30 states now.
So this is spread by fecal matter that contaminates foodstuffs. And so the reason why, as you mentioned, some of these herbs, leafy kind of herbs that are affected is because they're somewhat harder to wash. So just gently washing them you can't get all of the material off of the material because you got crevices that are there that kind of prevent that. Or if you get a spring mix of vegetables, like washing it is a little difficult to get every leaf of the salad mix clean. So that's why in the fresh kind of leafy vegetables, you're having this problem. If you cook the food, so if you put the cilantro in something and you cook it to 158 degrees.
But then that would be,
yes, no one does that. So good on your if you do, that kind of then will kill it. You said onions. Remember, onions have a skin on it. So most of the time you will peel the onion.
Yeah,
You're not eating the onion skin.
I don't think onion is
so that's bad, that was my thing. That it's mostly just these leafy kind of vegetables that you have.
Like the bag
salad mixes. That's
really...
right. Yep, because it's hard to wash those. You can't get the leaves washed up. So basically,
for the last week, I've been avoiding fresh vegetables, just in general.
Right, that's what I'm saying. You can eat most of the vegetables and stuff are good to eat. You don't have to worry about it. And even if you get this, it's unpleasant. But if you stay hydrated, you're probably, if you're in good.
And then that's the thing. It depends on who gets this, how bad it affects you. But also, okay, so then.
I'm gonna die, guys.
What'd you say?
Yep.
You said, well, shit, I'm gonna die, guys. I've been eating a lot of greens lately.
I said, you're probably okay. So
we have a garden that we haven't harvested a lot yet. We're still waiting for that. But if I go out and get the cabbage leaves and wash them. Okay, so is this something that is going through the water? So you go out and you water your garden?
Fecal contamination.
But how is it getting on these plants? How is it getting on this produce?
On the field.
Yeah, it's...
And then, is it transmitting from a person to another person?
So it doesn't move from person to person. It's not like a norovirus or a stomach bug that like a viral or bacterial infection that does go from person to person, poor hand washing, and all of a sudden you get it, or a buffet line. This isn't that. And so you're right. So it is in the water, but that's it's contaminated water with fecal matter. So agricultural water, field workers contaminate the water that they're using for that. And so it always goes back to farms. It traces to human sanitation breakdown. So if there's some sewage leak or some problem, then you have that. The water that we drink and the water we're probably watering our plants with out in the gardens, they are treated.
So is this something like kind of like a recall? Like a lot of times you'll get a thing that says...
They will recall the foods that potentially this happens with, and if they're monitoring properly. And then you asked endemic and pandemic, just to circle back. Endemics usually are localized. You can trace it, you can map it, you can predict it. Pandemic is like the scale changes where it's more global and it's no longer a local kind of phenomenon, but it's more global now. So this here, maybe endemic, 31 states or so. So it's kind of...
So something's going on if it's happening in that many.
Well, our industrial food complex.
And people getting, and regulations going down.
I'm not going to speculate on everything. Yes, food regulation can be a problem.
That relates to science, and science not getting paid for, so then people not finding out. Okay.
So yeah, leafy stuff, grown in your own garden, you're probably good to go. You don't got to worry about anything. Go to local farms, the farmers, what they're doing. Probably good. But yeah, industrial farming complex. That's one of the problems, especially if, like you said, regulations are lax, anything like that. And yeah, things like this can happen. So interesting.
It's always rough for everyone.
It is. Yep. I didn't really know a lot about this organism until I started looking it up a little bit.
So there you go. Thank you for that science hole.
Yep. Hopefully. But don't freak out.
Science hole.
You can just cook it. You're okay.
Yeah, just learn to cook. It's always a good thing.
Clean it, prepare it. But yeah.
What about you, Joe? What do you got for us this week?
Yeah, so I had a couple things, and one was on CRISPR. So CRISPR always comes up. We're trying to plan an episode for season 4. But one of the cool things, and I've talked about this, is the epigenetics. So epigenetics is this: you have your DNA, which has instructions, what genes to turn on, turn off, to do different things. But these are chemical groups, methylation groups that sit on top of the DNA to actually have another level of control that is applied to the instructions of the DNA. And these things they're hereditary, so they can be passed on. They can be induced by stress, by poor diet, by your environment. And so you can imagine that once you have an epigenetic change, that this change then will persist through generations. And so one of the things, and these epigenetics, they can turn things on, off, maybe things that's supposed to be on, it turns it off. So this can lead to health kind of consequences. And so what they're doing, they're saying, well, can we use CRISPR to modify the epigenetic coding that is applied to the DNA? And so there was a study, a couple of research studies, companies, startup firms are coming out where they are developing therapies that specifically target the epigenetic marker. So not changing the genes where CRISPR cut and paste, but actually changing the chemical modifications that's put on the different genes. So really interesting research, and kind of you can go in, you can treat things like high cholesterol or rare muscular disorders. These kind of companies are going after that are controlled, that are caused because of these kind of epigenetic patternings that are being applied. So that was really interesting and tethers into the episode with some of the things we were talking about, genes turning on and off, and how would you make a minotaur, I think was one we kind of chatted about. And then the other one that was interesting, once again, epigenetics. And so it was a paper, the gut bacteria epigenetic failsafe. And essentially what happens, you have a population of bacteria. They're all individuals, all single cell individuals.
but they
want to hedge their bets, right? So if the environment changes, they want to make sure that they are able to withstand that as a population, maybe not as an individual, which is really interesting. And so some of the members of this population will use epigenetics to turn on or turn off different genes so that if their environment changes, antibiotic comes in or the environmental nutrients change, that even though you might wipe out much of the population, some of it may still live because it made these kind of epigenetic changes that it then can modify to actually persist in the event that something like this happens, a catastrophic environmental event. So that makes
basically that's evolution.
Well, kind of. You're not mutating genes to change the gene product. You're changing the modification on top of the gene product. So it's a little different than evolution.
It makes me think of that, though, because I can always think of the one thing that I remember, the moths in a certain area, they were like, do you know what I'm talking about? They were like gray.
England, during the Industrial Revolution, the moths were white. And then when you had the soot built up, the birds could eat the white moths because they could see them pretty easily.
Or some gray ones or some
gray ones started. So there were gray ones in, and this is that the phenotype. So you always had probably white and gray, let's say, moths flying around. The white ones just for whatever reason. So then you change their environment, and now the gray colored moths had a selective advantage. And then the white ones were eaten at a higher rate, and then the gray ones could multiply at a higher rate.
Their genes were more...
That's right. So their genes became more persistent in the population than the white ones. Maybe the white colored ones didn't go away because it wasn't, but you still had this.
When you were talking about what you were talking about, that's what kind of came into my mind.
I see that, yeah.
Yeah.
But so in that case, right, so these bacteria, it could be, if there was an epigenetic change right there where you were a white moth, but then you changed, you got stressed, you saw your buddy get eaten, then all of a sudden you go, oh man, you methylate then your gene, and it goes gray.
I have to think, this is probably thinking about this on way too simple terms or practice, but like you were saying, trauma or stress can cause these things. And you said that can cause certain genes or certain things to turn off. So let's say you're in a program where it's to reduce stress and you're using meditation. And then that meditation would that be considered what you're talking about?
Yep. You could. I mean, potentially. Yeah.
Because I feel like that's such a bigger practical thing, and then you're talking about something on more of a cellular level.
Yep.
But that really
that's exactly
is.
It. Yep. That's right. Your body responds to that. This peaceful environment. You can think of it, kids who grow up in very stable, quiet neighborhoods versus kids who grow up next to an industrial factory.
Or dangerous.
Right. They're going to have the stress, their environment will cause them to have, their body's going to respond to that at a cellular level. And then you go, oh, why are these kids sicker? Or why are they not as healthy? Or why can't they concentrate as well? Well, because their body made these changes to fit that environment, not fit this environment now that's over here. Right. So, But yeah, so they're trying to now, can you use tools to undo that? Interesting.
That's wild. I didn't expect them to be able to do that.
Yeah, no, that field's moving pretty fast, and they're going after taking these tools. And
I honestly feel like every time I'm just so shocked by what it can do.
Yep. So, but yeah, so that was a couple there. And then the tether into this infectious disease model, the people were talking about the public health systems in the 2026 World Cup that's happening right now. But they're saying how that creates the perfect condition for infectious disease spread, right? So you have all these people coming together.
Because there's so many people.
People coming together and then migrating, right? Because you might go to a game in Kansas City and then you travel to LA or Philadelphia, the sites, Miami. And so you get this mix where and then you go to Canada or Mexico. So you have this kind of
perfect storm.
Right. So you could have stuff, you could have potentially.
Don't eat the cilantro, guys.
Right. So they do the tabletop modeling. You might have heard when we had Charles Blue on talking about for planetary defense, but you can do the same thing for disease modeling. And so potentially have outbreaks of measles, influenza, mosquito-borne viruses. So you can have maybe at the high end of risk would be things like Ebola, and they limited travel from countries where this current Ebola outbreak. And so they're actually denied or quarantined before they can come in, because if they get into these populations, you could have a disaster, a disease spread event. So,
but they let the team in. Do they have to do a special test?
They probably did containment. They probably containment or
they did during COVID time.
Yep. I don't know if their team was in. Sorry. Do you know that?
Oh, I don't.
Oh, okay. No,
I don't
know. Their country might not have had a team in. Not every country had a team in the World Cup. So, but yeah, so that was, I thought that was interesting and tethered into what you were talking about, the explosive diarrhea.
Yeah. We all love to talk explosive diarrhea here at Rabbit Hole of Research.
I know. That's it. You gotta go.
It's not a pleasant rabbit hole.
No, that was
pretty sure that's going to be our most popular mini.
I know.
Just so we talk
that's
about
it. Let's put that in SEOs. But, yeah, so those are a couple.
Very good. You guys want to jump into what media we've been consuming?
Yeah, let's do it up. What do you got?
I think we're calling this section our media hole.
The media. We start naming
the media holes.
All the media holes. What are we wasting time on?
The media hole over there.
Yeah. What long format?
Yeah, because
unprocessed media you've been enjoying.
The holes that I found myself falling into are the stupid scrolling on my phone.
Right. The ultra processed holes. We want to. No, everyone can do that. Yes. It's just like eating food.
We try to get out of the doom.
Yep. Like you eat food, you got to mix it up. You can have chips and ice cream. Good stuff, but you got to have a well-balanced diet.
That's all I've been eating lately, because it's not leafy green.
Wash your vegetables. There's other vegetables you can eat. You don't need to.
Nope, George is out. She's just one.
Yeah. We should have mentioned that there, but berries too, like things like that have nooks and crannies, right? Yeah, that's right. They're hard to clean, so if you get splash on from the water, you get or cook them.
If you do get berries, use baking soda. Oh. It's always a good thing
to wash them with.
It's a kitchen hack.
Yeah, nice.
Just let it soak for like 15 minutes
in a watered baking soda.
Yes.
Okay.
Yeah.
Nice.
Yeah, that's always a good thing to do with it. If you have littles, it helps kill a lot of the bacteria and stuff that's on it.
Yeah.
Yeah.
But you got to remember, that's for bacteria. This here is a protus, the Cyclospora. So it has a really thick cell wall that protects it. That's right. So I think that's what makes it so difficult. People are treating it like it is a normal bacterium or kind of cleaning it in normal food preparation.
That's why it's scary.
This isn't that. So you have these, like the vinegar, lemon juice, even bleach might not be as effective on these organisms.
Raspberries and bleach.
So it is
you don't like giving your raspberries and bleach, Georgia? Yeah. I love just drinking the bleach.
Yeah. And the baking soda is probably a little more for pesticide removal and things like that. I think folks use it for that. But yeah, for this, it is kind of...
you should be cleaning them, yes.
Yeah. You should clean, but you do have... It's difficult because of the structure of the leaf or the berries, because they have like little grooves that the oocytes of this parasite can get into.
And this thing is resistant.
That's right.
To
and
that.
it has a big cell wall that makes it hard for things to get in. That's why it's tough to kill. It's like Steven Seagal, hard to kill.
So on that note, let's move into the media holes.
The media holes. I'm going to change my title on this now.
Do you want me to go?
Sure. Let's do it up.
So,
go right ahead.
and this is actually a segue for a really nice plug that I can do. But my friend Jim and I are big fans of film noir. You might know if you've listened to the podcast. And we have a calendar that will be our 2027 calendar, and it has lots of movies, noir facts. And one of the movies that's mentioned in there, I realized I haven't watched this movie, and I've heard of it several times, so I thought I better watch that. And it's a 1951 movie called Ace in the Hole. Have you guys heard of that? And it stars
No, I can't say I have.
A very young Kirk Douglas. And the funny thing is, Eddie Mueller, who's the expert on noir, feels like it's not a noir because a lot of it happens during the day. I don't know. So it could be more noir adjacent. But I'm telling you right now, you need to watch this movie. It is the starkest, it has this commentary about media that I've never seen before. It sticks with you. So basically, Kirk Douglas is this real cocky young guy, and he's this journalist, and he doesn't necessarily, and it's really poignant right now because the whole idea is about truth and journalism and media, and he happens upon this small town hunting for Native American artifacts and this cave-in, and he's down there under some rocks, and it's how they deal with getting him out of there, and it becomes the media circus. That's what happens, and basically it's how can we make money off of this person's horrible situation. But you really I recommend this movie. Highly recommend it. It's not an uplifting movie by any means, but it makes such a great point about how we exploit people and things and how we get caught up in these. And I just think about social media and media today, and it's just so relevant.
Mm-hmm.
Yeah, so I highly recommend that. And then the other book I read, The Wolfworm by G. King Fisher. And it kind of goes right with the parasite.
That's right, yep.
Yeah, because it talks about a parasitic insect, and it's set in like 1899. It's from a woman's point of view. She's a scientific illustrator. And it really talks about naturalists and the study of nature. And so I think it does actually have quite a bit of science in it, too.
Well, yeah, the screwworms, they're flesh-eating insects.
Yeah, it's
worms, yep.
That's terrifying. So,
those can get wild. Yeah.
So it is a horror book, but so good. It's so well written. I love that.
Yep.
So that's
I'll
me.
check that one out. That sounds really good.
Yeah, I think you'd really like it.
Yeah, I think we mentioned it. It's a little squirmy. It'll get under your skin, literally.
Literally, yeah.
Figuratively, hopefully not. Literally, yes.
Squirmy, wormy?
Yes.
Yeah.
This worm will literally get under your skin. I hope not.
Very good.
But, hell yeah.
Yep.
What about you, Joe?
Yeah, so I'll just go 2 books. One's fiction. I started listening to Pimp My Airship by Maurice Brodus. It came out in 2023. It's an alternate history tale, kind of revolution and resistance, steampunk, Afro, sci-fi adventure
and was
story.
Was that someone that you met?
I did meet him. Yep, I met him at Con, Carolina.
And
so Annie lives near Indianapolis, I believe. And this story is set in Indianapolis area, so it's really interesting. So I'm about halfway through. So
and
I'm really
I saw him at the ALA conference.
Right. Yep, that's what you were saying.
Yeah.
Yep. So, and then the other book I'm reading is a nonfiction, When Trees Testify, Science, Wisdom, History, and America's Black Botanical Legacy by Breonda L. Montgomery. And this came out months ago. And so she is a PhD plant biologist and explores the way, in particular, 7 trees and cotton shrub are intertwined with the Black history and culture. And the idea is thinking about how the knowledge surrounding the trees, they shaped American kind of history from the beginning of our time here in America as Americans. And so, and you think of some trees live hundreds of years. So what memories do they have of some of the events?
And that really makes me think of Charles Chestnut and the Conjurer Woman.
Right. Yeah.
And he had a story about the trees on that one plantation had been chopped down, and it was like they were using it as lumber, and like and it actually held some of the spirits and
No, it's interesting because some of it plants they use CO2 that we exhale, and to use that to make sugars and for them in the process of photosynthesis. And then they expel plants oxygen that we can use and we can breathe. So it's an interesting thing because people that are around, if a tree lives that long, then you could say if this person was around this tree, then some of their CO2 they exhaled could become part of this tree and their sugars and their bark that they use and make complex structures with those sugars. So yeah, it's interesting.
That's right. Yeah.
And then I mentioned a children's book where they were making chimeras, like a hig, which is a horse pig that says oink neigh. It's called
with an oink neigh here and an oink neigh
Young McDonald by David Milgren. And so it's a really fun book. It explores genetics in a very fun way by reformulating the classic Old MacDonald jingle song. Yeah, it's really fun. Young McDonald. So, okay. That's it for me, my media hole. What about you, Nick?
Oh, yeah. Well, I have been playing an older game lately. Not all the way done with it, but a good chunk of the way. Watch Dogs 2. It is about hackers and stuff. And the first one takes place in Chicago. The second one takes place in, I want to say, San Francisco. And yeah, it's just a really cool version of what goes on with tech and the dangers of it. Just because anything you could do online, it's there for everyone. And it's just like it's a good analogy of what's going on with the world and stuff. But I've been playing that. And, what's the name of the show? Been watching an Apple TV show with Titiana. Do you know what's her name? One who played She-Hulk.
Oh.
Is this the one Maximum?
Yes, Maximum Pleasure. Guaranteed.
Yeah. I haven't watched it, but I heard it was really good. And I know they've been really talking about how, because I think Stephen King was recommending it.
Yeah, I thoroughly have been enjoying that show. Yeah, I've been watching that and just finished Widow's Peak.
Oh,
or Widow's Bay?
Widow's Bay,
Widow's Bay.
Widow's Peak.
I love Widow's Bay.
Yeah, that was really good.
That was so good. I guess they came out with an extra episode. Did you hear about this? I haven't.
No.
I haven't looked at it. It's not like a regular episode, but it sounds like it's almost like a collage of different things that make, just like the mood of Widow's Bay. It was supposed
This isn't like the Netflix thing, Stranger Things, where it was like, oh, a new ending episode.
I think this really is a thing. I have to look.
Okay.
I hope not.
Because it was like...
If it's like that, I can't watch it.
Yeah.
Because it was like, hey, to hold you over until we come out with another season.
Right.
It's like just the ambiance of Widow's Bay.
What people remember when Stranger Things, the series finale came out, and then people was a little disappointed, a little flat.
Some people weren't. I liked it.
Yeah, they said there was another version that was going to drop, and they gave a date and a time, and then Netflix crashed because everyone logged in at that. Yeah, so, okay, that's different.
No, I'm pretty sure this was, but you're right. I don't know if I really read about that on a very official...
reputable source.
Right.
So
where was it at?
I'll have to do some research.
I am on Apple right now, and I only see 10 episodes. Nothing more than that.
Oh my gosh. I might have been.
You've been duped.
I thought of something else that I didn't mention. I also this weekend saw Obsession, and I have to say, we don't need to talk about this movie because the billion other people on TikTok and Instagram talking about this movie, but it holds up to the hype. Let me just say that. Have you seen it?
Yeah.
Nick?
No, I have not.
I haven't either. I haven't seen it.
I don't go to the movies.
I know. I know.
I got a toddle.
I know. I'm just saying. A lot of times when you hear this big hype and expectations, you go in and then it's hard to live up to that. No.
I do get that 100% because a lot of movies that get super hyped for me, I'm not the biggest fan of.
Yeah, I know.
It's like...
That does happen to me quite often, but this stayed true to what people said. It's intense. And it's very different. And I think it's just so exciting that a 26-year-old director is making this movie. He made it for $1 million.
Oh, yeah.
And I
would highly recommend checking out his comedy stuff, too.
I don't even know if this number probably isn't even accurate anymore, but the last thing I saw, it was made 200 times its budget. 200 times. So you don't have to have a huge budget. You know what I mean? You can be young, you can be innovative, and I think it's like...
definitely showing that the indie space has what it takes for it. You don't have to have a huge budget for horror movies.
No,
horror movies just need to be good.
Oh my God.
Like it shows that.
And the acting in this movie, if there aren't some awards come out of that, that'll be like a shame because that... anyway, you gotta see it.
Well, on the opposite end of that spectrum, I watched a bad horror movie at home. I did watch the Return to Silent Hill, and it was a rough watch. I watched it all. I thought for a minute that they were doing some stuff intentionally, like everything just had a video game look, but it was just, I'm pretty sure it was just supposed to be bad CGI. But it felt like it was early 2000s.
Because that was a video game, right?
Yes. And this was an adaptation of Silent Hill 2. And it's stuck very true to the game for the most part.
So does that mean you'd recommend? Would you recommend it? Or?
I'd recommend the video game, which they had re-release of that come out last year, I believe. Yeah, that sounds right. And that was a fun replay. But the movie is a rough recommendation. Georgia, maybe you guys should watch it next week and let me know about it.
See what happens.
See what you
I'm going to watch bad movies. There's a lot of good stuff.
I can see if I can pencil that in.
Yeah, you got some time. Don't worry. And then one other thing I'd like to bring up is that someone on our Discord had messaged me and they sent me some of their artwork, and I just wanted to give them a little shout out. Lorene Doodles. Yeah, they had some really cool stuff, and we're about supporting the small artists and shit like that.
So maybe we can put a link. Can we put a link in our show notes?
Yeah, sure.
Yeah.
If I remember to send Joe it tonight, yeah.
Yes, please send it. We'll get it. If not this one, the next newsletter. Every Wednesday, a newsletter goes out, so we can get it into one. Mm-hmm.
Very cool.
So
nice.
You just go check it out. Tell them that we sent you over here.
Yep. Cool.
Got anything else for us, Joe?
I think that might be it. I was going to mention I was at a con, the Shore Leave 46 con in Lancaster, PA. And one of the things was I went to a panel. It was an adaptation of Fallout. And so it was a really interesting discussion. And the thing came up about the video game. And they're like, oh, Joe, you got to play this video. You got to play it. So I might see if I can. Which I'm surprised because it came out in 97. And that was probably when
actually play
I was playing a lot of video games, especially on the Doom, that kind of descent right in that time frame. So it's really interesting that this must have fallen right into where that gap between where I was playing a lot and not playing a lot, because I was in grad school at this particular time. So yeah, very interesting.
Yeah, you should definitely check out the game.
Yeah, but it was a good conversation. I really enjoyed it. But yeah, it was a good con. I gave a talk about the electron microscopy of zombies and cells. Well received. Maybe I got to post some of that on there. But yeah, cool. Yeah, but that's it for me.
Whoa.
Yeah,
cool. We went down quite a few holes.
So mini is going to be...
we go down all the holes.
We're going to have to change... We're going to have to change...
The maxi.
We're going to have to change the name.
Maxi Mini?
Yeah.
We're going to have to change the name of this.
Maxi Mini hole. Yeah.
The mini squared hole. But yeah, I think that's it. Thanks for listening. Thanks for supporting.
Yes. And don't forget to tell all your friends. And
friends,
give us
you got a question?
High five.
You got a question or something you want us to look into? Let us know.
Science hole you'd like to
go
down?
We can do it. Might not come in the next episode, but we can put our heads together and figure it out. But yeah, thank you.
We do try to get to everyone's messages and stuff. It just takes time because we are humans with other things that we do.
We got stuffs. But on that note, you got me, Joe.
You got Nick.
We got Nick.
Georgia.
We got Georgia.
And down any hole.
That's right. So stay safe out there. Stay curious. And we love y'all.